Hello, this is Emilio. We're going to be talking about retroviral vectors. So there's two main families for the retroviral vector. There's the orthoretrovirinae and the spirometrovirinae. Most studies are done on orthoretrovirinae. So under that subfamily we have alpha, beta, gamma retro, delta, epsilon, and lenti. Most of the studies are done through lentivirus, gamma retrovirus, and alpha virus. And we'll see for some of the reasons why right here. But in general, for all retroviral vectors, integration is pretty much the virus inserting its genome into the host. And once that process is complete, it's established in its host, and it just becomes a part of the host. But there's some differences between each sub family. Like gamma retrovirus, it's used in a lot of studies. However, it's dependent on the nuclear membrane degradation, so it could only affect during cell division. It doesn't have an active mechanism to get across the nuclear membrane. Whereas on the other hand, we have the lentiviral vectors, where they could actively pass the nuclear membrane, and that's it makes the process of transducing cells more efficient, where it's not dependent on the cells dividing. So looking at the parts of a retrovirus, the genetic information of it, it's a single-stranded RNA, and at both ends we have long terminal repeating sequences. And these are necessary for the integration because they act as promoters and they bind with the integrase protein. And if we look in between those LTRs, we can see there's a portion called GAG. GAG is responsible for encoding structural glycoproteins, and it's required for the assembly of non-infectious and immature viral-like particles. And right next to that, we have the POL, which encodes enzymes necessary for viral replication and integration. So stuff like protease, reverse transcriptase, and integrase. And right next to that one, we have the ENV, which encodes proteins that are embedded in the viral membrane, which plays a role with recognizing cells. This is not uh, true for all retroviral vectors, but this is just a general single RNA strand, and which is kind of overlays with each one. There's some variations though within different subfamilies. So looking at the enzyme integrase, it's what integrates the viral DNA into the host genome, and like I stated before, it's encoded by the pol gene. Once it's actually produced, it assembles with DNA and cellular proteins to form the pre-integration complex, and this complex is gonna associate with the host DNA and complete the integration process. So looking at the integration, I'm gonna break it down into three segments. I'm gonna quickly look over the structure of integrase and how the integrase finds its location where it's gonna integrate the viral DNA, and then I'm gonna go over the mechanism of integration. Here we can see a 3D picture of the integrase protein. It's a tetramer, a symmetrical tetramer. This picture also includes the viral DNA bounded to the integrase. On the right picture, you can see in purple and orange the viral DNA, and those are the two ends of the viral DNA. The inner proteins of the integrase are responsible for all contacts, and the exterior proteins act as more of a stabilizer for the protein. So focusing on the integration site on the host DNA, one study focused on the frequencies near CP islands and also the start of a transcription site. So first, looking at the key that we have here off to the side. In red, we have the HIV virus with no substitution of different proteins from a different virus. In yellow, we have a HIV virus, but the GAG gene was replaced with a murine leukemia virus GAG gene. And green, we have again the HIV virus, but now the integration is swapped out again with the murine leukemia virus. And in blue, we have the HIV virus, but the GAG and integration was replaced. And in the violet purple, it's the appear murine leukemia virus with no HIV association. So with the pure HIV, that acts as a random integration site. And in purple or violet, it's since it's using the all of the viral proteins from murine leukemia, it's going to be more centered to these sites. And looking at the results of their study, notably, we can see in green and blue, these have both of these virus, these chimeras, they have the murine leukemia integrase incorporated in it, and we can see the influence of that. We get a stronger frequency of integrase towards the center of these CP islands. And as expected, our murine leukemia virus is showing high frequencies, and our HIV pure virus, it's randomized throughout this CP island. And looking at the integration site near active transcription sites, again we're getting similar results, except the green and blue, the integrase and integrase GAG replacements, they're showing strong integration towards the center. And again, with the HIV peer, it's randomized throughout the sequence, and our murine leukemia virus is showing strong integrations towards the center of this transcription start site. The main takeaway from the study is that integrase plays the major role in where the viral DNA is going to be integrated in the host DNA. Other viral proteins can influence, but integrase is the main one. You could take an integrase from one virus and replace it for another virus integrase, and it's gonna show the same integrating patterns as the host or the original virus. So looking at the integration mechanism, it's really understanding the mechanism underlying retroviral integration, as well as identifying positions in the host genome where retroviral vectors integrate, are crucial to further improve retroviral vectors for safe gene therapeutic applications. So breaking down the integration mechanism down to 4 steps. So first we get the formation of the nucleoprotein complex, which is just getting the two ends of the viral DNA, and it's going to associate with the integrase. Those are going to be fixed inside the integrase, and in between the two inner proteins of the integrase, that's where our host or the host DNA is going to be associated at. But with the host DNA, it's going to be bent to an extent that's going to be exposing the phosphodiester bonds within the DNA. So there's two catalytic activities for the integrase. It's going to be 3' processing of the viral DNA, and then the DNA strand transfer. So what happens is with the viral ends, they are cut by the integrase, exposing the 3' hydroxyl group of the viral DNA, and it's going to use those hydroxyl groups to cut the the phosphodiester bonds of the host DNA. And so when it completes that cut, it's also integrate. It forms a new bond with the host DNA, successfully um, transferring the viral DNA within the host DNA. And then it's going to be the host enzymes that complete the process. So it's just going to be filling in missing base pairs from the viral DNA when it got cut by the integrase. And that's the citation. Thank you.